folding a couple of questions together for Dr. Bazhenova. So we've talked about kind of in the earlier segment, biomarker testing, right? Next generation sequencing to help kind of elucidate who might benefit from a targeted therapy. Where do things stand with respect to antibody drug conjugates? Can we kind of learn anything from our next generation sequencing? And then to tack on a second question that might be semi-related, someone in the audience astutely noted that we've recently been we've seen a bunch of press releases of failed studies in comparison to docetaxel. How do we move the field forward? Any insights as to why are not, perhaps strategy, study design, et cetera, why those studies may not have been successful?
Yeah, so docetaxel has been our nemesis. We hate it with a passion because it does have pretty significant toxicity. We are really working hard and find something better, but so far everything kind of fails over the docetaxel. I think, it's hard to understand why antibody drug conjugates over docetaxel trials, at like the recent press release of CCAM trial, and were negative. I think the question is, should we be using the biomarker targeted strategy, right? So technically we are developing antibody against, TROP2, right? Why are we not testing the patients for TROP2? And there is a reason why we are not, is we've done that. And we saw that you see responses pretty much regardless of the TROP2 expression. And so maybe TROP2 is not the right biomarker. Maybe it's a biomarker of how good the ADCs are being engulfed by the cancer cells. It's actually also interesting for the DATA-DXd, which is datopotamab deruxtecan, the recent release of the second line troponin, I forgot the number, I think it's 05 study. They showed that there is a definite improvement in non-squamous non-small cell lung cancer, but lack of benefit in squam. What to make out of that, it's really, we don't know. There was a question about, can we use next generation sequencing to decide which patients will benefit from antibody drug conjugate and which of them will not? I think it's a little bit too early because yes, you can find the TROP2 overexpression by RNA, but the studies were not designed to test the strategy, and we can certainly do that. I still, honestly, even though the randomized studies against docetaxel didn't show what I wanted to see, to me, I still find the value in antibody drug conjugates because it gives me an extra treatment option. Even if I give patients docetaxel, the median progression-free survival in second line is 4 months. So what am I going to do afterwards? And give gemcitabine, and the median PFS is like 3 months. So it's like the more treatment I have in my bucket, then I think honestly I'll be a little bit better off. And then toxicities are certainly slightly different as well between those two, and maybe you can win on toxicities in a way.